Hi everyone, my name is Zhan Chen, and today I'm excited to present our recent work on a fine-tuned dataset and benchmark for large language model for protein understanding. This work is a collaborative effort involving TourSynbio and several prestige institutions, including the Johns Hopkins University and the Shanghai Jiao Tong University. Now, let me introduce our work. In recent years, significant achievements have been made in natural language processing through large language models, which are trained on vast datasets to handle complex language tasks. Inspired by this breakthrough, our research addressed a fundamental question. Can large language models originally designed for natural language processing be adapted to understand protein sequence? This is crucial because protein sequence, like natural language, follow sequential structures, and a deeper understanding of this sequence can greatly benefit fields like medicine and biotechnology. However, existing datasets do not adequately bridge the gap between protein sequence and corresponding textual description. This limitation hindered the ability of large language models to fully comprehend and utilize protein data for tasks such as protein functionality prediction, disease involvement, and more others. To address this gap, we introduced two new resources: the Protein LM dataset for large-scale pre-training and fine-tuning of large language models, and the Protein LM Bench, a benchmark designed specifically to access protein understanding capabilities in large language models. The Protein LM dataset is the largest protein text hybrid dataset designed to empower large language models for both self-supervised and supervised fine-tuning. It includes over 70 billion tokens for pre-training and 893,000 supervised instructions. The pre-training dataset is divided into three primary segments. The first segment is Chinese-English text pair for protein science literature. The second segment is protein sequence and English text pairs. And the third segment, we called it protein sequence collection, but it actually is we insert protein sequence into English text. Each segment contributes uniquely to help large language models learn and understand the complex relationship between protein sequence and textual description. The first segment of the dataset focuses on Chinese-English text pair in protein science. We extracted 7,000 entities from scientific literature, including journals such as the Chinese Journal of Biotechnology. These pairs contribute more than 120 million tokens and are particularly valuable for cross-lingual models. Given that both protein science and NLP are global fields, this dataset segment ensures that large language models can work effectively across different languages, a crucial feature for collaboration and research at the international level. The second segment in our dataset focuses on protein sequence and English text pairs. This process is more straightforward, involving the pairing of protein sequence with relevant scientific literature. We used PDB IDs and UniProt IDs to create mapping between protein sequence and their corresponding research articles. Here's how we construct this pair of the dataset. Firstly, we obtain the protein sequence information from CIF database, which provide mapping from PDB IDs to UniProt IDs and subsequently to the protein names. This allowed us to identify articles related to these proteins from sources like PubMed and PMC. For each protein sequence identified, we gathered full text article or abstract from PubMed, which describes the biological role, structure, or function of these proteins. The dataset gathered from this process include around 80,000 entries, providing a subsequential foundation of over 195 million tokens. Moving to the third and one of the most critical segments in our dataset, we focus on the protein sequence collection. Unlike the last segment, which was centered around pairing protein sequence and existing literature, here we delve into a more advanced process where we extract named entities using advanced NLP techniques and map them to their respective protein sequence. To build this dataset, we applied BERN2, a state-of-the-art named entity recognition model, to identify key protein and gene mentioned from PubMed articles. We proceed the title and abstract of PubMed articles, extracting nouns that could refer to protein or gene. To ensure quality, we assigned each extract entity a confidence score, allowing us to focus only on high-confidence extractions. The next step involved named entity normalization, where we mapped these entities to their respective entities in the NCBI gene database. This process gave us a precise gene or protein ID for each identified nonce, enabling us to move forward with the collection of corresponding protein sequence. Once the NR and NN steps were complete, we utilized NCBI RefSeq database to retrieve protein sequence that matched the identified genes. At this point, we have both the protein sequence and the corresponding entities mentioned in the literature. Our final step involved integrating this sequence back into the original scientific text. For each mention of a protein in the abstract, we inserted the protein sequence immediately after its first occurrence in the text. This not only created a cohesive dataset where textual descriptions were directly paired with protein sequence, but it also provided a detailed context for training models to understand how these sequences are discussed in scientific literature. This process allowed us to generate a rich, contextually relevant protein sequence dataset that directly links scientific discourse with the underlying biological data. Additionally, the supervised fine-tuning dataset consists of 893,000 instructions across 7 tasks, including protein functionality, post-translation modification, and disease involvement, and so on. One key contribution is the introduction of enzyme chain of thought, we call it ECOT, which enable large language models to think step by step in protein science, enhance the reliability and accuracy of their outputs. This approach marks a significant leap forward in protein science comprehension. To evaluate the effectiveness of large language models in understanding protein data, we developed a protein LLM bench, a benchmark dataset of 944 multiple-choice questions. These questions were initially generated by GPT-4, which created questions, options, answers, and explanations based on randomly selected protein literature. We then reverse validated each question by re-inputting the generated questions, options, and explanations into GPT-4 to ensure consistency. If the answer matched, the question was deemed valid. Finally, protein science experts manually reversed the dataset with only a small number of questions removed from potential issues. We believe that the protein LLM bench was a new standard of evaluating LLMs in protein science. In our experiment, we trained the InterM2 7B model on both self-supervised and supervised data from protein LLM dataset. We then evaluate the model's performance using protein LLM bench. Interestingly, the model trained on the full dataset achieved an accuracy of 60%, outperforming even GPT-4. This demonstrates the potential of specialized dataset in enhancing LLM's understanding of complex protein information. Our results clearly show that Training large language models on protein-related datasets significantly boosts their performance in tasks such as protein functionality prediction and disease involvement analysis. This opens up new possibilities for automating protein sequence analysis and applying AI-driven tools to synthetic biology and medicine. However, there are still limitations to be addressed, such as potential bias in dataset correlation and the technical challenges inherent in accurately representing protein structures. In conclusion, our work presents two key contributions to the fields of protein science and AI: the creation of Protein-M dataset and Protein-M bench, which together form the first large-scale dataset and the benchmark specifically designed to train and evaluate LLMs for protein information understanding. These resources pave the way for future research and combine natural language and protein science, unlimited accelerating advanced in biotechnology. Thank you for listening. Our dataset and benchmark are publicly available on Hugging Face, and the code can be accessed via our GitHub repo. I'm happy to take any questions you may have.